In this slideshow, we're going to be looking at what DNA is and what it does, kind of briefly explaining the process of gene expression and looking at some of the causes and effects of damage done to the genetic code. So as we think about the age in which we live, it's sometimes referred to as the age of the gene. It seems like gene therapy, stem cells, cloning, forensic analyses, parental determination, everything and every facet of our lives, genetically modified foods, all comes down to knowledge and information about the gene. And so it's important that we as citizens understand at least a little bit about what this means and how genes function. So there are two important features to know and understand about DNA. First, that it contains all of the genetic information about an individual, how to create the organism and how to control its growth and development. It also is a set of instructions that are passed down from parent to offspring. So these are heritable features that can be inherited from one generation to the next. James Watson and Francis Crick are often the two men credited with having discovered DNA, how it's structured. But truly, Rosalind Franklin was another key piece of the team to discover and unravel, if you will, the secrets of DNA. Watson and Crick established the, at first, hypothesis about the double helical structure, but Rosalind Franklin was one of the first to confirm that, and you can see on the image on the bottom right this X shape, and this is an X-ray diffraction of a chromosome taken by Rosalind Franklin. When Watson and Crick won the Nobel Prize, I can't remember which year it was, but when they won the Nobel Prize, Rosalind Franklin would have been a portion of the team to receive that also, but the Nobel Prize is a prize that cannot be won posthumously, and she had already passed away. So looking at this DNA and the double helix, we looked at last week, DNA can be seen as a ladder rung, and that's what you're seeing on the bottom left-hand portion, with a sugar and phosphate and sugar and phosphate and sugar and phosphate molecule on either side, but one is inverted from the other. So where we have sugar and then phosphate, on the other side we have phosphate and then sugar. For DNA, that sugar is going to be deoxyribose. That's going to be the pentagonal sugar. And attached to this sugar and phosphate backbone are going to be a series of nitrogenous base pairs. And there are 4 bases that are used in DNA, the A, T, C, and G, adenine, thymine, cytosine, and guanine. 1 phosphate group, 1 sugar, and 1 nitrogenous base are going to make up a nucleotide. And in DNA, we have a series of nucleotides double-stranded, and in RNA, we have the series of nucleotides in a single strand. And the base pairs involved in RNA are going to be almost the same, with one exception. We have A, U, C, and G, the same adenine, the same cytosine, the same guanine, but uracil is present in RNA. We had looked at this last week, but go ahead and try this just to see if you remember, with from one side to the other of the double helix of DNA, which would be the base pair to match that sequence. Just unpause the video when you're ready. And hopefully you realize that this should match up with number 3, that the A should bond with T. A does not bond with itself, so you can rule out the first 2. And in DNA, we don't have uracil, so that leaves 3 as the only option. So genes are portions of the DNA, and what they're going to do is they're going to code for everything, every facet of every organism on the planet, is going to have DNA that's going to code for making the proteins that are going to make up the cells and make up everything about that individual. It's what's going to make fish different sizes, what gives them the different scale colors that they have. It's what enables lily pads to be differentiated from grasses, from shrubbery, and from trees. It's what makes each bird unique, and so on as you go throughout an ecosystem. DNA is going to code for RNA, which is going to code for proteins, and it's going to make each organism different from the other organisms around it, and each individual unique. So as we look at this, we start out at the genome, and the genome is the complete set of DNA for an organism. And there are going to be some differences, but generally we can talk about the human genome, and all human beings, regardless of gender, regardless of race, are going to have just about the exact same genome. This has been established. What of the 23 chromosomes that we have, what is located, what protein codes for what function on each chromosome is the same for all human beings. And that's what makes our genome. And this DNA is found in all of our somatic cells, every single cell, with the exception of sex cells. So with the exception of eggs and females and sperm and males, every human cell has DNA that codes for the same genome. The chromosome is circular in prokaryotes. In linear, we generally depict it as these X's. That's when the cell is going through its cellular division. Otherwise, it's usually one line. And for human beings, we have 23 of them, 23 chromosomes, which are going to be made up of hundreds of millions of base pairs. Chromosomes join together to form the genome. Smaller than the chromosome level, we have specific genes, and that's going to be one segment of a chromosome, usually about 3,000 base pairs. So if you take the hundreds of millions and divide by 3,000, you see that there are a large number of genes that are going to make up each chromosome. And each gene is going to code either for the production of a particular protein or one portion of a particular protein. And so DNA codes for RNA, and RNA is going to code for proteins. We're going to start out with a genome, and that is the entire set of genetic information that all human beings have and all organisms have. Then we're going to have in humans 23 chromosomes that each contain a couple of hundred of million base pairs that are going to code for a series of proteins that make us uniquely human. And then our genes are the subunits of a chromosome, and it is at the gene level that you're going to find differences, and that's how each human being can be unique. We can have different heights and different colors and all of the variation that makes a human being. And these differences are going to be found in alleles. Alleles are different versions of the same gene that code for a different trait. So the combination of the protein, the genetics behind it, the individual base pairs that are going to make it, combined with some of the environmental factors, are going to make different versions. And so we have all of these horses are the same species, but we can get a various variety of traits. The genotype is what the genes actually say, and we'll see this in a later section that we use capital and lowercase letters. So we can have two capital Ds, and that's going to give a phenotype, that's the physical expression of a chestnut. We can have a capital D and a lowercase d, and that's going to be palomino. And if we have two lowercase d's, then that's going to be this, I don't know how to pronounce it, cremello. The chestnut is a different gene, and so when we combine those two, we can get very similar styles of horses, very similar colors, but sometimes the tail and lower portion of the leg are a different color. And so the genotype is what literally is in the genome, what the order of the base pairs are, and the phenotype is what we see as a result of that, what the physical expression is. And again, these are called alleles, the different versions of the same gene that are going to code for a particular trait. So if you look at these, take a moment and fill in the blanks. Okay, and hopefully you got that each of those is a gene and the two different versions are alleles, so number 4. So I'd mentioned this before with the horses, but the genotype is literally all of the genes, each segment of each chromosome that's going to make each organism unique. And the phenotype is the physical manifestation of it. So the genome, at the genetic level, we're not going to see that, though we can test for it. But the phenotype is what we see. That is a short woman with hazel eyes and brown hair. You don't see the genotype that's going to build it because you don't look inside of my blood and inside each cell at the DNA, but the phenotype is the physical manifestation of it, and you can see all of those physical characteristics. And two important processes are going to determine how cells, or not determine it, but they're going to enable cells to share this genetic material outside of the nucleus where it's stored. And the first portion is transcription. That's where the DNA is unwound and a section of it is copied so that we can make a particular protein. So that's where the DNA becomes RNA, transcription that takes place in the nucleus. Then the RNA can leave the nucleus and the DNA will wrap itself back up and stay in the nucleus for protection. The RNA leaves the nucleus and goes into the cytoplasm, and that's where translation occurs. And that's where the RNA is going to build the protein. And so we're going to look at these two processes, transcription and translation, in more detail. Sorry, and I should have said that mRNA, messenger RNA, is the segment that is copied. That's the portion that leaves out of the nucleus and goes into the cytoplasm. And tRNA, or transfer RNA, is the portion that's going to put the amino acids in sequence so that we can build a protein. So the mRNA is the strand that has the code off of the DNA, and the tRNA is going to carry the amino acids over to this production line where a ribosome is going to bond them together and form a protein. So now you can answer the question as which one functions as the middleman. And hopefully you realize that it's number 2, the mRNA, the messenger RNA, or you can think of it as M for middleman RNA, but really it stands for messenger. So the first portion is transcription. Again, this is going to take place within the nucleus. It involves the DNA. The DNA cannot leave the nucleus, so this whole process takes place within the nucleoplasm, or the jelly-like fluid within the nucleus. So the first section of transcription is going to be the unwind and then rewind part. That's where the DNA is going to literally unwind from that double helix, and an enzyme called RNA polymerase is going to move down the DNA. It's going to be unwinding it and enabling this process of production of the mRNA. That's this red tape-looking thing coming off of it. And what that is an exact copy, except that T's are replaced with U's, but a copy of the transcript of that one half of the DNA, so that that can go off, leave the nucleus, and code for the particular protein for a ribosome to make it. Once it hits an end, the RNA polymerase is going to break off, the mRNA strand is completed, and the DNA itself can zip back up and kind of rewind itself. So we have initiation, where the RNA polymerase attaches on. We have elongation, as the RNA polymerase moves down words across this section of the DNA, and the mRNA strand grows, and then termination, where it reaches a key sequence that indicates, okay, time to pop off, and that's when the DNA is going to zip itself back up. So the whole portion of transcription, the whole process rather, is going to be recognition and binding. That's when the RNA polymerase is going to recognize a promoter site. There's a particular sequence of base pairs that's going to say, you can attach here. The RNA polymerase will then attach there and will read down the gene's message. The transcription part is the DNA strand being processed. The RNA polymerase is going to build a single-stranded copy of that one segment, that either gene or that portion of the gene, to build the mRNA transcript. So this is the copy of the DNA, except where the U takes the place of a T. Then we get the termination part. It's going to hit another recognition site that says, okay, it's time for you to break off now, and the enzyme, that RNA polymerase, is going to detach from the DNA strand, and that's when capping and editing begins. Before the mRNA transcript leaves the nucleus, it's going to have a cap, essentially a protective protein, placed on either side, and all of the portions that are not involved in coding are going to be removed. So essentially it does, just like you would edit a paper, it will be edited to remove any non-essential parts of the DNA sequence before it goes out into the cytoplasm. So this is the transcription process, again, that takes place in the nucleus. So try to think about what we went over last week and match up the DNA segment with the appropriate mRNA segment. And hopefully you realize that it was number 2. The T will match up with an A, the C with a G, and the A has to go with a U because now we have mRNA, no T's in it. And translation is the second portion. This takes place in the cytoplasm, so the mRNA section leaves the nucleus and goes out into the cytoplasm, where it's going to be able to meet up with some essential components, free amino acids that have been recycled or broken down, either made by the rough ER or recycled from the lysosomes, ribosomal units. These are ribosomes that are not attached to the rough ER, and transfer RNA, and that's these things that have a little 3-prong appearance. All of these are going to be integral in the translation process in the cytoplasm. So transfer RNA is going to be the important component for translation. We had the messenger RNA that was built during transcription, and the tRNA, the transfer RNA, is going to be responsible for literally transferring each of these amino acids. Each of these 3 sets of base pairs are going to code for a particular amino acid. So those 3 are called a codon. The mRNA has a particular series of these 3 base pairs called codons that are going to call for a particular tRNA to bring a particular amino acid so that they line up in the correct order so that the correct protein is built. And if you remember, there are 20 amino acids that can build the hundreds of thousands of different proteins that we have in our body. So recognition and initiation is the first portion of translation. It's always going to start with an AUG. That particular sequence, that codon, is going to call for the beginning, and it's going to call for one particular amino acid, and you'll see that when we look at the codon chart later. The elongation part is going to be once the tRNA, two subunits for a ribosome can attach on at that AUG starting point, transfer RNAs, tRNAs, will come and bring particular amino acids so that ribosome can build a protein, and that's the elongation part. So we have this series of codons on the mRNA, and a whole suite of tRNAs are going to bring one after another amino acid so we can build this yellow string of proteins. Then a termination sequence will be met. This is a stop sequence, and there are different versions of stop sequences. They're always going to start with A, U, G, but the stop codon can be different. In this case, it's UAA that says, okay, we're done, that protein has been completed, and it signals for the two ribosomal subunits to break apart, and the ribosome and that section of mRNA can be reused. Once that mRNA has been used a series of times, then it will be broken up by the lysosomes and can be recycled. So this is the codon chart, and I will go over this in class, and so we'll go over this answer together in class. Now, how do we get differences, right? This is going to be integral in a few chapters when we get to evolution, but there can be changes in the sequences of bases in DNA, and these can lead to different structures being formed because different proteins will be produced. Sometimes this is a good thing, and sometimes it's not. So usually we associate mutations with being bad. Hutchinson-Gilford progeria is sometimes referred to as the aging disease. The person that you see in that upper left-hand image is actually a child of about 10. Usually people afflicted with Hutchinson-Gilford disease don't live past about 13 because they suffer from the effects of old age. Ectrodactyly is sometimes referred to as a crab disorder. People will tend to have only 2 digits. This can happen on the hands as well. We see a mutation in a frog here where it has 6 eyes, 3 different sets of eyes, and that was caused by an environmental mutagen. And Proteus syndrome is sometimes referred to as elephantitis, and this is the man who is referred to historically as the elephant man. So each of these is a mutation where a chromosome or a set of base pairs was not put together in the right sequence. So they tend to have a bad reputation, right? Mutations are not good things. They tend to be disruptive, but not always. Certainly in all the previous images, those were not positive things, but they can be positive, and they are very, very rare. Sometimes we can have positive mutations, like sickle cell. While sickle cell anemia is a terrible disease, it so happens that if you are a carrier for sickle cell anemia, you will be immune to malaria. So presence of the sickle cell can be a very positive mutation. And once a mutation occurs in an individual, it can be passed on to the next generation. So it's hard to say if this red coloration was good or bad, but if it happens to make you, sexier and more attractive to the other fish, then that might be a positive mutation. So they can be good, they can be bad, but in either case, they're very, very rare. And there are 3 different types of mutations. There are insertions, deletions, and substitutions. So if you look at the initial statement here, my daily bread, with a substitution, the A was changed to an E. With a deletion, you can see the B was left out. And with an insertion, the S was added. If they're broken up into triplets, then it would be read as the codon. And so you can see the result would be in the substitution, you have 1 bad codon. In a deletion, everything after that codon is going to be affected because essentially you've moved up each base pair by 1, and so all subsequent codons are going to code incorrectly, and the same with an insertion. Once you had this mistake, every single codon is going to code incorrectly. So in general, Substitutions are not nearly as detrimental as insertions or deletions. And so we see these represented again with the two basic types of mutations, a point mutation in which one particular base pair is affected with either the deletion, the duplication, or the insertion. You can also have an inversion where it's literally put in upside down, or a translocation where you have all of the genetic material and it's all present, but it gets put in the wrong place, and that's a translocation. So this can happen at a point mutation level where it's one base pair, or on a chromosomal level where the entire section of a chromosome, a whole gene, is offset. And again, with these as well, insertions and deletions are usually much more serious than substitutions because they affect a greater percentage of the gene. And the sources of mutations can be varied. We can have radiation, either natural from the sun or artificial in humans from X-rays. We can have chemicals. Again, they can be artificial, like everything listed here, or they can be natural, like the decomposition of radon in rocks. Or infectious agents are usually bacterial or viral. And you can see in this image on the right what the effect is on the DNA sequence before and after, where the mutagen has ripped apart this portion and damaged one section of the gene. So once this tries to copy, we're going to be looking at most likely a deletion where this entire portion will be absent from the next version of this gene because it's not going to be able to run through the translation or transcription process appropriately. So another one to practice. And hopefully you realized that once again the T's are going to have to go with U. T's have to go with A's and A has to go with U. So we're once again going to have number 2 here. And which one will be the most severe? Pause it and come back when you're ready. And hopefully you realized that number 3 is going to be the most severe because it is the only one where all the way up at the second codon it has been misaligned and replaced. So we have the second through final codons are all incorrect, so that one will have the most serious ramifications. Okay, and why are these mutations, or why can they be serious? If it's causing an illness or a genetic disorder, the gene that has the mutation is going to code for a protein or an enzyme. And because this is coding incorrectly, the wrong protein or the wrong enzyme is going to be made. Then, assuming that it was an enzyme, it's not going to be able to catalyze the reaction that it normally would. So if, say, it was the ability to digest lactose, now you can no longer do that. That function is going to stop, which means that that substance is either going to accumulate in the body long-term or short-term. In the case of lactose intolerance, it's fortunately short-term and it will pass. But in the case of Tay-Sachs, it's disastrous, and then either sickness like indigestion, upset stomach, and severe cramping and nausea and diarrhea can result from lactose intolerance, or death in some cases, as is almost certainly the case by age 1 with Tay-Sachs. So think about this one for a moment and unpause when you're ready. Okay, and hopefully you realize that unfortunately all of these can occur, and so a change in one nucleotide can lead to a cascading series of effects.